My name is Barbara Nowak and I'm the president of Australian Society for Parasitology. I had the pleasure to recently be guest editor for International Journal of Parasitology on the special issue, double issue on fish parasites. And I have here with me today Associate Professor Louise von Gersdorff Jørgensen from University of Copenhagen. And Louise published one of the papers in this issue. So Louise, can you tell us more about the paper?
Yeah, sure. Hi, Barbara. Well, we did this study on a parasite called Ictyophthirius multifiliis, which is a common freshwater parasite in aquaculture. And in relation to climate change, we wanted to investigate whether a change in pH would affect the parasite. So we measured size of the parasite and observed survival and if development was delayed or not when you change pH. So we kind of found out that pH does have an effect. From before, we know that salinity, for example, has an effect on the parasite, and the temperature can affect the size of the parasite and the length of the life cycle. And we found some of the same stuff with changing pH.
So with effect of pH and potentially temperature, do you think climate change will have an effect on the parasite itself?
Yeah, I think it will if pH changes and if the temperature changes, but since the parasite survives up to 30 degrees, it may not be eradicated, for example, in many areas. If, let's say, the temperature goes from 25 to 26 degrees, then the life cycle will just be faster and it will survive perfectly well because it is not very host specific. It can use many freshwater fish hosts.
So it's quite a significant issue for fish farming, for freshwater fish farming then.
It's a very significant issue. It's an economical, huge problem all over the world, actually.
So if it's such a huge problem, can you tell us more about the parasite itself?
Well, it's one-celled ciliate. And as I mentioned, it's not very host specific. And it infects freshwater fish on the skin and the fins and the gills. And if you want to, I can show you the life cycle and we can take it.
That'd be fantastic. Yeah, sure. I would really like to see it.
Here you have the infective stage of the parasite. It's called, this one little guy is called a therond. And the therond has 24 hours to find a new freshwater fish host. And then it will infect either gills, skin, or fins. And after a while in the host, let's say at 15 degrees, it may be around a week or 10 days. And then the fish will look like this with white spots all over. And it actually here looks like an ectoparasite, but it is an endoparasite because it's covered by the very outer layer of the skin, and it's a transparent layer. So we can see the parasite, but it's actually inside the fish. And then when the parasite matures on the fish, it will leave and seek bottom substrates. Actually, it will swim around for a while. You can see some of the tomons here. It's actually still one cell, but it's just a huge cell now. They will swim around and find a suitable substrate, and then the tomon will encysst itself, and now it's called a tomocyst. And within the cyst, a lot of tomites will develop, and the tomites will turn into therons, and and then the life cycle is complete. Over here on the video, you can see how therans escape the tomocyst. This is the whole tomocyst, and therans are escaping all the time. And so that was a little bit about the parasite.
It looks like a very complex life cycle with lots of different stages.
Yeah, but it's still direct. It only has one host.
And how does the temperature affect life cycle of egg?
When you increase the temperature up to around 30 degrees, you just speed up the process. So actually ornamental fish, like I have a picture up here of ornamental fish, they are also hit hard by this disease. So when you treat your fish, your ornamental fish, you are allowed to treat with some substances in the stores. They actually also turn up the temperature to kind of burn it out, to make the life cycle go really fast so they can treat for a short period of time.
That sounds really interesting, and it's like good to have options to manage the disease by using environmental factors. And you mentioned salinity can be used to treat some of the stages?
Yeah, mostly the free living stages, and you actually have to go relatively high, from 5 ppt to maybe 10. The parasite is not going to be very happy. But many things don't like salinities above 5 ppt. So yeah, that is all. Sorry?
Obviously, you have to make sure that the fish tolerates the higher salinity before you use high salinity to treat the parasite.
Yeah.
That's really quite amazing. Have you done other research on egg?
Yeah, I've been working with egg for quite some years now, and we have attempted to develop vaccines against egg, because you can actually say that it's a very special parasite, because you don't get hosts to obtain immunity against many parasites, but this one you do. So you actually have fish, when they have been infected once, they will most of the time acquire almost 100% immunity. And that provides us the basis for knowing that it should be possible to make a vaccine. So we have attempted this for many years. We have attempted with a DNA vaccine and a subunit vaccine. And well, that is not going to go on the market because it's not that effective. So we need to keep on going and include maybe more antigens and other ways of immunizing. So that's still ongoing work for sure.
But overall, it sounds quite promising. So maybe there'll be a commercial vaccine against fish parasitic disease in the future.
Yeah, there should be actually, because in theory, it should be possible. We just need the right tools to do it.
So obviously, when you were developing the vaccine, experimental vaccine, you must have looked at the host parasite interactions for egg.
Yeah, we have. We have done a lot of work in rainbow trout with gene expression studies and the measurement of antibodies. And we know that the host reacts when it acquires protective immunity. We know the host reacts with the Th2 response, producing a lot of antibodies. And actually, to more go into detail with cellular responses, we have used the zebrafish as a model to investigate immune responses against the parasite. And we have these very nice, oh, wait a minute, we're just gonna skip this. This is all about immunization. So we have these very nice transgenic zebrafish lines we can use. And this one has a green fluorescent tag on the neutrophils. So when we look at the fish, we can actually see how neutrophils move around in the fish. And since from histological sections, we can see that around the parasite, we see cells, but they are half degraded and no one actually knows what they're doing. We wish to investigate how neutrophils responded towards the parasite. And we wanted to investigate it both in immune fish and in naive fish. So this might be a bit of a complicated slide, but we'll go through it nice and easy. So the top row up here are control fish, which are not infected by the parasite. The second row here is naive fish, which are infected with the parasite for the first time. And here at the bottom, we have immune fish, which are infected with the parasite for around the 5th time or something like that. So they are really immune. And what you can see, all the green color, that is neutrophils. And these are adult fish, because you cannot infect smaller fish with ick, then they will die. And we have to look at fins, because as you can see here, if you look at the body, there are so many neutrophils that the whole body of the fish will just be green. And fortunately for us, the parasites do infect fins. So, so we took a good look at the fins. And here you have before infection, and this is 1 day after infection, and 2 days after, and 3 days after. The most interesting thing you can see here is that 24 hours after infection, we have these accumulations of neutrophils in the tail fin of actually both the naive and the immune fish. That was actually a bit of a surprise to us, because we kind of imagined with a strong Th2 response, maybe the immune fish wouldn't need all these neutrophils to attack the parasite when it invades the fish. So we also looked at, do the parasites really infect immune fish? And how long do they stay in the fish? So I will just take you to another slide here, because we have the naive fish up here. We have the immune fish down here, and then they get infected. This one. And you can clearly see in the naive fish that after 2 days, we have a lot of ick on the fins. And we can see in the immune fish, there's no ick. But actually after 1 hour, if you take a closer look, you can see ick on the tail of the immune fish. So if you watch in detail, you can actually see small white spots 1 hour after infection. So even the immune fish, they get the same amount of parasites as the naive fish. I have actually encircled them if they are too hard to see. But then after a while, they're gone. So 24 hours later, we don't see parasites anymore in the immune fish. But we do see a lot of neutrophils. So the question is, why are neutrophils so, why are there so many in a tail fin, which does not harbor any parasites anymore? And we also looked at gene expression, and normally, or many neutrophils are attracted by this chemokine CXCL8. And at this point, there's no upregulation of CXCL8. So there must be some other attractant to the neutrophils, because the number of them is significantly elevated here in the immune. So something else must attract the neutrophils. It's not CXCL8, which would be the normal scenario.
That's really interesting.
Yeah, it's very interesting. I know that it's been discovered in humans. That's another cytokine like IL-13 can also attract a subpopulation of neutrophils. So something similar might happen in these fish, but I don't know, and that would be very interesting to look into.
Is it possible at all that because you have the parasites there 1 hour after infection, even the fish can get rid of them, the parasites can cause some damage to the tissues? And that's what causes the neutrophil infiltration?
Yeah, there might be some damage, but the parasites are very near the surface of the fish, and that regenerates very quickly.
Yeah, that's true.
So yeah, of course, there might be some damage, and also maybe some parasite leftovers. Maybe they have shed some cilia or something like that, and we know that the surface of the parasite is full of antigens. So there might be something keeping the neutrophils there from the parasites. But we don't know. That would be very interesting to look into.
That's really fascinating. And most of this work has been done with zebrafish as a model?
Yeah, it's an excellent model for this. The thing that was not described in literature was how you make the adult fish lie still when you examine them under a confocal microscope and keep them alive at the same time. So
yeah. So that was the biggest challenge, was it?
Yeah, it was a bit, but we kept on going and found a way.
So this graph showing? Yeah,
this graph is showing because we counted the neutrophils as well, and the green stuff here are controls, and the black dots are naive fish, and the blue ones here are the immune fish. And you can clearly see that we have a higher amount of neutrophils in the immune fish, even compared to the naive fish. And that is when it becomes very puzzling to us, because what is the sense of that? The naive fish should, in my opinion, react with a stronger neutrophil response than immune fish, because immune fish have antibodies. But we looked into this as well. So this is a picture of the same tail fin, just before infection, 1 day later, 2 days later, 3 days later, and with normal light, so we can see how the parasite grows. And with GFP light, so we can see the neutrophils. Because we wanted to know in more detail what was going on when neutrophils were attracted to the parasite. And as before, you can see 24 hours later, after the infection, there's this huge accumulation of neutrophils. And then the number of neutrophils go down. And that is what was really weird, because the parasite just grows and grows and grows. And when it's this big, it is doing a lot of damage. It can actually move around underneath the epidermal layer. And it is eating a lot from the fish, and it's really destructive. So why does the number of neutrophils not increase instead? We took a closer look at this and spent a lot of time video recording parasites on the fins in these transgenic fish, where you can see the neutrophils like this. This is the parasite, and it's gonna rotate when I turn on the video. This is another parasite. This one is moving out of the picture. But to see exactly what happened between the parasite and the neutrophil, so you can really clearly see how the neutrophils are moving around. It's quite amazing. And what's this neutrophil? It's coming up towards the parasite. So it looks like it's sensing the parasite somehow, and it's projecting these pseudopods towards it. But the parasite might also sense the cell, because it's gone. It's gone. And
Wow.
that is, that can explain a lot of the things we saw, because we saw that the number of neutrophils in naive fish was less than in immune fish. But that makes sense if all the parasites in the naive fish are just eating all the cells. In the immune fish, the parasites are kicked out. So all the neutrophils can accumulate with no problem. But in naive fish, they get eaten.
Wow, I think it's really fascinating to watch.
Yeah, but there are more cells. It's been eating more neutrophils. And in all the parasites on the fins, you could actually see green spots inside. They fade relatively quickly, which means that they disintegrate inside the parasite. But in all the parasites, I could see these green spots moving around inside. So we kind of knew what was going on, but we really wanted to film it.
Yeah.
So it also explains why the number goes down over 1 and 2 days, like we saw on the previous picture.
That's really amazing. This is really fascinating. So you're doing research with very sophisticated tools. As you know, usually fish parasitology doesn't use sophisticated tools, but you are looking at zebrafish as a model and having fish which have fluorescent neutrophils. It's really sophisticated. So have you thought about using zebrafish as model for other fish parasites, or do you think ick is very important parasite and you should focus on ick?
I think I will always kind of focus on ick because I've been working with it for so long and know it so well now. But the zebrafish can be used as a model for many parasites, in my opinion. Unfortunately, something like salmon lice is probably not an option because that's a saltwater parasite. It's very important in aquaculture. But I don't think we can use it because zebrafish, they are not gonna last. They can maybe take 10 ppt, and that is not good enough for salmon lice.
So most likely very good model for freshwater parasites.
For sure, a perfect model, actually.
Yeah. So what are you planning to do for your future research? How are you going to follow up these amazing achievements?
Well, I would really much like to do the same kind of study focusing on macrophages because we are very focused on immunology, and macrophages are kind of one of the links between the innate immune response and the adaptive immune response. And we actually have the transgenic lines in the lab with the fluorescent macrophages.
Oh, fantastic.
So we can do it when we kind of get the funding for it. But that's definitely a wish. That's something I'm gonna try to achieve, to do it somehow, because I want to see what's happening with macrophages.
And possibly develop a vaccine.
For sure. That's also going to be an ongoing thing until we have one, kind of. So a vaccine would be so valuable all over the world.
Yeah, that's true. So good luck with the funding and congratulations again on your paper in IJP, and hopefully we'll see more results soon. Thank you.
Thank you for including me in this interview. I'm always happy to talk about the parasite.
Thank you.